All right, okay, ayan. So hello dears, hello. How are you all doing? I hope you're all doing fine and nag-enjoy sa screen break. Yes, kamusta naman atong screen break kahit 1 week lang. Ako mismo nag-screen break din, no. Nag-detach ko sa akong self, nag-detach ko sa social media and all that. So thankful po ko ato na screen break. But like sa duration sa screen break, ang nasa akong mind kay akong na-feel kay murag nahuman na ang semester. And then what I was supposed to do ato na week kay mag-calculate na grades and then and all that. But yeah, sige na lang. Let's just libre lang mangarap. Malapit na rin naman matapos ang semester, di ba? But anyway, yes, I hope you had a fruitful and very enjoyable na screen break. I think some of you should really enjoyed, no? Sana all. May mga punta na dito sa Duma. Sana all. So welcome to Duma once again. And I just hope again that you all took care of yourselves and really you were all careful ha. So ayan, laban lang. But anyway, okay, so welcome to this parang just a further elaboration no, of your immunochromatography because I remember during our pre-recorded lecture katong sa YouTube, the one that I uploaded in YouTube, I mentioned that I will include parang an example talaga no on immunochromatography so that you will really understand how it is done. Okay, and I mentioned about pregnancy testing. So because again, it's one of the most popular na mga kits na ginagamit by people, which is again pregnancy testing. And I believe some of my students already now in IS have been introduced to this method because some of them again were my students sa AUBF last sem, analysis of urine and body fluids. So this is just a review, hopefully. Okay, and for the new ones, then I new students of mine na karon pa lang naka-encounter ani sa pregnancy testing. Then I hope magets lang yun ang overall principle and how immunochromatography works. Okay, all right. So we'll start first with this slide, diba? So this was the structure or the picture of your kits. No, what are the components? So you have of course a sample port, but the name itself diri mo ibutang imong sample. And once your sample is dispensed here, it will then be absorbed by your sample pad. And then of course, diba, recall that ang flow sa imuhang imuhang assay is padere, 'di ba? Okay, that's why it's also called as lateral flow, 'di ba? Naka-recall ba sa lateral flow immunoassay? Okay. All right. Ayan, because again the flow is padulong here from sample pad to the absorbent pad. Okay. And then of course, so after the sample pad, after the sample is being absorbed by the sample pad, it then goes to the conjugate pad, the membrane, and then the test line, control line, and finally the absorbent pad by the name itself to absorb the excess reagents, the excess sample. Okay? Alright, so that's basically the structure inside your kit. Okay? So, bahalag maingon mo na, grabe ka simple naman yung test kit, pero the principle is not. Okay? So, you have to, um, you have to be familiar. Okay? So, medyo wrong spelling pala ito. Immunochromo, wrong spelling. I'll edit this. Immunochromatography. Okay? Medyo na wrong. Okay. Alright. Ay, sorry. Ayan. Okay, so again, 'di ba, for the overall flow, again, the first is the sample is loaded in the sample pad. And then, of course, ang movement man nato is again from one to here, diba? So, the sample then, again, goes to your conjugate pad, which again reconstitutes the labeled antigen or antibody, depending again on what you want to detect, okay? And then, of course, they then form a complex that migrates towards the detection zone, which is either the T or C, okay? And yeah, an antigen or antibody, again, depending on what you want to identify, immobilized on the detection zone, meaning naghulat na siya dito, wala na siya nag-move. It's there. It's adsorbed there. It's immobilized there. No? Naghulat na siya dito. Sana all willing muhulat. Charot. Jollibee kasi. But anyway, all right. And then they form an immune complex and then form a colored line for a positive test. And then finally, of course, excess labeled immunoreactant and the sample also migrates to the absorbent pad, which is number 4. Okay, so that's basically parang a rough overview of how immunochromatography works. Okay, but for the next slide, we'll then look into an example good, which in this case is the one-step pregnancy test. Okay, and what we are detecting here is, of course, recall in your AUBF, sana naman, I remember, is your beta HCG or beta human chorionic gonadotropin. Okay, all right. So for the procedure, ayan, so it's of course your test kits are usually sealed. They are found in sealed pouches. Okay, so usually we we store them inside refrigerators together with their reagents. Okay. Um, so before we test your kits, we need to, of course, let them go back or let them uh, be at room temperature. Okay, so after they are already at room temperature, we then open the pouch, remove the test device, and then place on a flat, dry surface. Usually on our countertops at ang lab activity, lab rooms would suffice. Dito ta mag-test. Okay, and then in this case, since this is pregnancy testing, what we are testing is again beta HCG, and our sample is urine. So for this, again, test, we use urine. So there's a dropper that's included in the pouch. So we use the dropper to again get our urine. Again, what is the most prescribed specimen to detect pregnancy testing nga? Na-remember pa ba? Sana naman. Ako mga previous students, dapat press the buzzer na. What is, again, the prescribed specimen for testing to avoid false negative? Ayan. Pregnancy test results, your first morning urine specimen. Why? Ano ganito? Because your first morning urine specimen is considered to be the most concentrated. Ayan, the most concentrated sample because it's the urine that accumulated overnight. Okay, sa pagkatulog nimo. So tanan gyud na sediments or chemical substances found in your urine, makoncentrate gyud siya, 'di ba? Kay first morning. So it's the first urine that you void or that imuhang i-urinate pag mata jud nimo. So it's really concentrated. That's why we want that sample to avoid false negative results, especially for pregnancy testing, because your beta HCG in this case is very concentrated. So, makoncentrate siya, ma-detect siya sa imuhang test. Okay? So, di ba? Nako, sana naman, press the buzzer na. Okay, all right. Ayan. So, you drop the urine into the dropper, using the dropper into the urine well, and then the positive result is a purple color band. So on the test line and the control line, diba? Okay. All right. And we interpret the result at about 3 minutes. Okay. And then again, the principle is immunochromatography. HCG, if present in urine, it will react this time with, ayan, mouse anti-beta HCG, monoclonal antibody. So therefore, there is an antibody that is waiting already, okay, in your excuse me, conjugate pad in the conjugate pad. Ayan. So it's combined with a gold colored conjugate so that it will be stable. Okay. All right. And so that it will be immobilized. Okay. All right. And then once HCG is present in your urine, it will combine with IgG, sorry, anti-beta HCG antibody, 'di ba? Kay anti-beta HCG, so specific dito siya to HCG. So they will combine, okay? And then this complex, they will form a complex. They will now move to the test region where in the test region, there's already nag-wait na po na antibody to HCG. Okay? Kaya iyahang i-capture, iyahang kuhaon tong complex na na-form. Okay? And then, same po sa same po sa control line. Okay? Which contains a goat anti-mouse IgG. Okay, alright. So, to really understand it even better, we'll have again another illustration. Again, this is from Sir Jemu pa rin ha, the best Sir Jemu. Okay, all right. So here is the sample kit no inside. So again, the flow of the assay is from the sample pad until to the absorbent pad. So that's why it's also called lateral flow immunoassay, if you can remember, diba? Okay. Now, again, in the test line, you already have an antibody that is waiting there. By the name itself, it's GOAT anti-HCG. So it's an antibody to HCG, okay? And it is derived from a goat, meaning ang goat gi-injekan niya og sa manufacturers og HCG so that the goat will produce antibodies to HCG. Because ang HCG sa body sa goat will serve as an antigen. Okay? Munang sa inside sa body sa goat mo produce siya antibodies to HCG. And then katon antibodies gikuha or gi-purify, gi-extract muna to ang reagent na gigamit. Okay? So sa test line mo ni ang gawit, you have a goat anti-HCG. And sa control line, you have a goat anti-mouse IgG. So it's an antibody against IgG. Okay, so it's an antibody against an antibody. So parang ganun. Okay, so there are antibodies already that's waiting on your test line or control line. Okay, now if your sample contains HCG, so the HCG then will serve as the antigen this time in the test system. Okay, siya ang antigen. All right? So pag butang nimo sa sample pad, okay, 'di ba? So diri man ang motion, 'di ba? Padiri. Okay? So this HCG, which is your antigen, 'di ba? Recall that ay sorry. Okay. That your conjugate pad contains mouse anti-beta HCG monoclonal antibody. So meaning these are antibodies to HCG nagikan sa mouse. Okay? So the next thing is of course if sorry. Okay. So the next thing is, of course, diba, the HCG will then combine with your mouse anti-beta HCG if it's present. Okay, so they form a complex. Now this complex will then move. Okay, all right, diba? So again, they will form a complex. Then this complex will then move padulong diri. Okay, all right. And of course, i-capture na siya with first sa imuhang test line, which is your GOAT anti-HCG. So, di ba recall that ang naa sa imuhang test line is GOAT anti-HCG. So therefore, it's an antibody that will react to your HCG. Okay? So gi-capture man siya, okay, sa imong GOAT anti-HCG. Okay? Kagets na mo sa procedure. So this is enzyme, di ba? So pag add sa substrate or sa imuhang reagent sa imuhang test kit, it will then result to a color band, a purple color band on the test line, which would indicate na positive siya. Okay? Di ba? And of course, the control line, it will then capture, okay? It will then capture the remaining of these reactants na unbound, okay? Kani mga gold colloid conjugate, okay? It will then capture diri sa imong control line, okay? Munang pag-add gyapon sa imong substrate ani nadyo'y color sige ang control line, regardless if nabay HCG or walay HCG. Okay? 'Di ba? Always gyud na in a test kit there should always be a control na appearance, 'di ba? Because you cannot interpret a result using a test kit if walay control na band. Okay? Because that would mean that invalid imong result. It means that your reagents are not working properly. It means that this is not uh, properly working. Because why would this result naa pero kani diri wala? Okay? It could mean that this is defective. It could mean na um, kaning antibody nagulat diri kay defective pod, so you cannot interpret a result using a test kit if ang control line wala. Okay? There should always be a control line regardless if in this case atong example na HCG which would indicate a positive result in ani or walay HCG which would indicate a negative result. Okay? Alright, ayan. And of course, the excess na mga reactants na wala nakuha sa imong antibody na nag-wait sa imong detection zone kaning sa test and control line will then be absorbed by your absorbent pad. Okay? So I hope na gets siya. So let's say ang another situation is what if negative? So let's say wala siya HCG. Okay? So what happens is of course, pagbutang sa urine, so ang movement is padiri, all right? Okay, so walay complex na ma-form ani niya. Okay? So therefore, sa test line, walay mo capture, wala siya ma-capture na ani na complex. So therefore, walay color na mahitabo pag add sa substrate, 'di ba? But sa control line, since namang gyapon ning excess na mga reagents, all right? So makuha ra gyapon siya sa control na antibody na nag-wait there. Kaya it's an anti-mouse IgG man, anti-mouse IgG. So same here, mouse anti-IgG or antibody, 'di ba? So iyaha japong dakpon. So therefore pag add sa substrate mo color gyud japon ang control line. Okay? That's why pag color sa control line nya walay color diri, 'di ba? That means a negative result. Okay? All right, generally in most of your test kits, but na ubang test kits na lahi iyang procedure like ay lahi ang interpretation like sa drug test na kits. Okay? All right, but that's generally the interpretation or the principle then of your immunochromatography kits. I hope na gets lang dears. Okay, I hope na gets lang yun. So I included this slide in your these slides. I included these slides in your handout para at least namoy ka na yung example gyud ba of immunochromatography na kit para mas magets ninyo. All right, but again that would kani mga reagents diri that would depend kung unsay mo detect. Okay, let's say kung antibody imong if antibody imong gi-detect here, let's say antibody to HIV, antibody to COVID, let's say. So therefore, ang naa diri pod is antigen na this time and ang gawit pod diri could be antigen pod. So lahi siya, depende kung unsay gi-detect nimo and depende pod sa manufacturer. In the insert, di ba recall insert kato ng paper na included sa mga test kits nato in our videos, sa mga lab activity videos nato. In the insert mismo, ilan nag-explain dito kung unsay mga reagents na naa. Like, is it mouse anti example kani mouse anti-beta HCG ba? Is it goat anti-mouse IgG. So, naka-explain na siya dito. Okay? So, it would depend. Naka-explain na siya dito. Again, depends on the the reagents would depend on the manufacturers and also on the sample or analyte that you want to detect. Okay? So, I hope na gets lang ni Dears ha. So, this is just an additional lecture on labeled immunoassays, specifically on test kits, okay, immunochromatography, so that you will understand what happens inside the test kit. Okay, so again, I hope na gets lang, but if you have any questions, again, as always, feel free to chat me or chat sa atong group chat. Nako, dapat di na dyan mo maulaw unta. Dapat di na dyan mo maulaw kay hapit na mo man ang semester niya magula-ulaw pa mo. My gosh, medyo mong others. But anyway, all right. So I hope na gets lang again, and I'll see you in our next pre-recorded lecture, dear. Hapit na gid mahuman. And yes, laban lang jud dira no. I think medyo naka-adjust naman mo sa third year. But again, under normal circumstances, unta gyud face to face, third year would have been such a memorable year unta gyud and one of the best. But sige lang, sige lang. Yeah, so wow, nagdrama, nagdrama, nagdrama ng konti. But anyway, yes, laban lang you dears, no. I'm supporting and rooting for all of you. Kaya lang yun na, no. I got you always, char. So chika lang yun. If you have any questions or concerns. All right. So I'll see you in our next pre-recorded lecture, dears. Have a great day and keep safe. All right.